Okay, welcome everyone. We're going to go ahead and get started. We are at the top of the hour. I am Cheryl Rogers, Director of Marketing here at Golden Helix. It is my pleasure to introduce Dr. Heather Huson. Heather is at Cornell University in the Animal Science Department. Today she's going to present her research surrounding population structure and genetic improvement in livestock. Heather was one of our winners in our first research competition, which we hosted last winter. Along with the opportunity to present her work today, she also did receive a brand new Dell laptop, as well as a license of the Snip and Variation Suite software. Come December, we will host our second competition, so if anyone's interested, just make sure you sign up to receive updates from us. You can do that on our website. Just look on the left-hand side, and you can register for updates. I will go ahead and pass it over to Heather now.
Thanks a lot. I appreciate everyone taking the time today to hear a little bit about my research on livestock. One thing, if you have any questions, please feel free to enter those questions into the webinar question pane, and we'll try to get to those as many as we can at the end of the talk. Today, what I'm going to talk about is really my research focus is on trait association and population structure in livestock. And with that in mind, we'll be talking about thermotolerance in tropical cattle, which is a trait association study, as well as the African Goat Improvement Project, which will show a lot more about population structure. In the long run, just about all of my work, has the end goal with genetic improvement of livestock in mind. So we'll start right out with the thermotolerance in tropical cattle. I started this project when I was a researcher at the USDA, and I've continued this, and we finished up the first segment of this project this past spring with a published paper. And we're going to start with really what's the importance of thermotolerance in tropical cattle. So the main issues here are looking at milk production and reproduction, the negative effects of if cattle can't handle these warmer temperatures, they have decreased milk production and reproduction as well as some other factors. With that in mind, our particular project really looked at the phenotype of the slick hair coat. So this is a short, fine, sleek hair coat that's often found in these tropically adapted cattle. On the bottom are pictures of 3 different tropical cattle breeds that we utilized in this study, primarily the Senepol cattle as well as the Karora and the Roma Sinuano. In all of these breeds, the slick hair coat is the predominant hair coat that they get. Occasionally you do find a longer hair coat in these animals as well, and it's still segregating slightly, but for the most part this is a fairly almost fixed trait in these cattle. The objective for us was really to improve the diagnostic markers for this trait, for the slick hair coat, and to identify the causative mutation. So a little bit of background about this project is that research actually started on the slick phenotype back in 2003 with Olson and collaborators looking at basically conducting breeding trials. Through these breeding trials, predominantly on Senepol cattle with a few Roma Sinuano, as well as crossing these to Holstein and Angus cattle, they were able to determine that this particular phenotype has a dominant mode of inheritance and was thought to have basically a single major gene responsible for the slick phenotype. They progressed with this study to 2007, where they did a genome-wide linkage analysis. So they used a lot of the breeding trials they worked on, and they were able to locate this phenotype down to chromosome 20. And they also determined that basically they had an area of interest from 37 to 40 megabases on chromosome 20, with a smaller conserved haplotype that was kind of their prime area between 37 and 38 megabases. They basically generated 3 microsatellites that are currently used as diagnostic markers for the slick phenotype at this time. In 2007, a French group went on to look more at signatures of selection, particularly in the Senepol cattle, and they came up with a region very near this, so closer to almost to the 39.5 megabases, using IHS and RSB as signatures. So they were interested in that particular part and looking at the ancestry of Senepol cattle. So to date, this was the research that's kind of come forward in this particular project, looking at the slick hair coat and Senepol cattle. So our role was really to basically improve the diagnostic marker, the 3 microsatellites, particularly one of them has proven a little bit difficult to utilize, as well as identify hopefully the causative mutation. So my role in this research was really to look at breed relationship of these different tropical breeds and ancestry, and look at the ancestral origins potentially of the slick hair coat, and also to conduct the genome-wide association analysis of this phenotype. So the research approach that we used was that we were really looking at using a variety of analysis. So it was myself and particularly one other colleague on this particular project, Dr. Isu Kim at Iowa State now, that we were looking at runs of homozygosity, signatures of selection, genome-wide association study, as well as haplotype blocks. So putting a number of different analyses together to see if they pointed to the same region. So a little bit about why was it important to look at the ancestry of these animals. So all of the tropical cattle breeds that we were looking at, and predominantly we had a larger sample size of Senepol cattle, is that these are composite breeds. So as a composite breed, the Senepol was developed in St. Croix Virgin Islands, and it's basically a composite of red poll and Dama, as well as we found, particularly from the French study, East African Zebu, and they probably also had some mixture into the local Criollo cattle that were on the island to begin with. So this comes into play because in general, the slick phenotype is going to be shown in almost all Senepol cattle, so we needed some type of reference or comparison population that didn't carry the slick phenotype, as well as playing a role in what were the ancestral origins of the slick coat. So we started out our work with kind of a genomic verification of that Senepol ancestry. So this is a diagram of admixture analysis. In this case, each vertical line is an individual animal, and we have them color-coded, so each different color represents a different genomic population that's brought out in the admixture analysis. And in this case, we're using a K=4, so there's 4 populations that are expressed here. The breeds that are represented in this particular analysis was starting with the Senepol because they were our target population, but then using the Ndama, the red poll, and the East African Zebu. And the main thing that I want to point out is you can see in particularly the more purebred individuals, like the Ndama red poll, that you can see that it's predominantly a single color of those, showing their more pure ancestry, whereas the Senepol in particular have a mottled coloration of the red color, which is unique to Senepol, but they also show varying degrees of the purple from Zebu, the blue coloration from the red poll, and even the green from the Ndama. We can break this down and basically look at what is the average genetic composition or breed composition of Senepol cattle. So if I take all of those animals that were seen individually and I average them together, I have basically about a 64% of their genome is significant and represented, correlated with the Senepol breed themselves, that unique combination of all these breeds together. But then we have about a 23% signature that's still defined as the red poll, and then there's smaller degrees of the Zebu and the Ndama, but they are still seen in each of the Senepol. So this basically verifies that as far as the Senepol, we can use these as ancestral breeds for comparison. So this brings us to one of the main roles that I worked on in conducting the genome-wide association study. So as I mentioned, this trait's generally fixed in Senepol, so we couldn't just use Senepol. We can't compare 36 Senepol that have the slick hair coat to only 2 animals that don't have the slick hair coat. And part of this is smaller numbers. We had more Senepol to choose from, but they were related, so we had to break these down to the least related animals that we could choose. So how we controlled for this is we basically brought in then the 10 Ndama, 10 red poll, and 10 Zebu as non-slick controls for comparison. But we had a number of other animals to access for this study as well. So from the earlier breeding studies, we had Senepol Angus crosses and back crosses, and now there the slick hair coat was segregating, so we had a few in each group. We also had the slick-coated Romo Sinuano, so only a few of those, but we had crosses between Romo Sinuano and Angus. So these are the slick hair coat with a few of them in the non-slick, but then we also brought in non-slick Angus as controls. We had Holstein Senepol crosses, and this was segregating in both sides, as well as we had non-slick Holstein to compare this with. And lastly, we had the third tropical cattle breed, the Carora, and their ancestry, which is non-slick, is the brown Swiss. So this basically gave us 72 cases compared to 61 non-slick controls. So the first analysis was basically done just using that minimal correction using the pedigrees for relatedness. And we at least see that we have a significant region on chromosome 20, which is where we wanted to see it. But you can note that there's a lot of background noise, a lot of SNPs that are still showing potential significance here. I have the black line at a negative log 10 p-value of 5 just as a reference for the next slide. So what I did here is I brought in, instead of doing kind of more straightforward GWAS, now I've employed the Emacs kinship matrix. So along with using pedigree correction for relatedness, I also used the kinship matrix. And this really made a big difference in showing what are actually significant areas versus non-significant. And you can see that at least in this case, the good news was that our peak on chromosome 20 was still there. So going on, we actually had a number of other analyses to conduct as well, and we wanted to see, do these same analysis basically have show us the same regions of association. So Isu Kim ran the runs of homozygosity for this particular analysis, and he looked at multiple different SNP thresholds. So when he did the runs of homozygosity, he had windows of 100 consecutive SNPs, 200, 300, and 500. And over our area of interest on chromosome 20, all of these different thresholds had the same peak, slightly varying degrees of significance, but they were all over the same area. Another part that we did was I basically broke this down and I looked at haplotype blocks with LD right over this particular region of interest then where our association was. And we found 24 significantly associated haplotype patterns. But one of the things, so in the table that you see, the very first one, block number 94, that happened to be the most significantly associated haplotype pattern with the SNPs there, what the actual allele calls were, and their frequencies in slick versus non-slick. But the ones that we found almost even more interesting were 3 different haplotype patterns that were associated, but in this particular case, they had no frequency. They were never present in these patterns in the non-slick animals. So these could be potentially diagnostic markers for the future. So a couple of the other things that we looked at for this project was looking at variation in genetic breed composition of the Senepol cattle. And so one way I showed you that idea of how we could look at the average breed composition of Senepol, and this was a slightly different way. So we took that same idea of the average breed composition of Senepol, but instead of just doing it on a genome-wide scale, which is normally what's done and what the previous photos were, this particular one, we're comparing how does that breed composition change when we compare genome-wide versus just chromosome 20 versus about 30% of the chromosome surrounding our slick region of interest. So with that in mind, you can see how if you look at the genome-wide, which is the top bar, you have, primarily this very unique signature to Senepol cattle as a composite breed. But you can still see the very small signatures of the ancestral breeds. As you narrow it down to chromosome 20, specifically near the slick region, you can see how the ancestral breeds, because they're showing basically more genetic breed composition here, how those breeds might, it makes you wonder, how do those breeds affect the slick phenotype itself? So that was, a way to kind of look at this a little bit differently when you're looking at breed composition. Another thing that we wanted to compare, since we were looking at kind of ancestry, was looking at the average breed composition of the 3 different tropical cattle breeds. So we really had the majority of the animals were Senepol of origin, but we also had the Carora and Roma Sinuano. And we do have to note that in this particular figure, we only had 2 Roma Sinuano as opposed to 10 Carora that we averaged together and 38 Senepol. So there's a very different degree of the animals represented. But in this particular case, you can see that the Roma Sinuano have a very unique breed signature, as well as the Carora. Each of these breeds does show some common genetic similarity with the others. And the Senepol actually gave us a unique signature where they do have 49% are the Senepol breed itself, but they have 25% that's genetically similar to Roma Sinuano, showing a more closely related relationship between Senepol and Roma Sinuano versus the Carora. So out of all of our different analysis that we conducted, we basically took those couple megabases that were the original area of interest for the slick phenotype, and we narrowed this down to approximately a half a megabase. So there's a number of lines here. This is basically a file from UCSC output, and I have inputted a number of different runs. So the green line at the very top was the initial 2007 area of interest. The purple line that's the next one down is from 2012 from the French group. And then our area of interest, the main area, was pretty much between the two red lines, vertical red lines. But you can see that the varying different analysis show us, you know, slightly different patterns. We have in light blue the runs of homozygosity. In gray, we have the entire GWAS region, and we have some haplotype blocks that are based on both Karora and Senapol that are in between in the oranges and browns. And at the very bottom, we have our three main candidate genes that we're interested in, looking for the potential mutation and causative gene of this slick phenotype. So this is where we've kind of brought this particular analysis to. Some of the outcomes were that we really did look at the breed relationship and ancestry of the slick-haired tropical cattle. We were able to narrow the locus to about a half a megabase on chromosome 20, and we did identify some new diagnostic markers, potential haplotypes that are not found in the non-slick individuals. And this we just published this past March in 2014. But we're not done there. So just some future directions. As I showed you from some of the breed relationship, we feel that there's a couple different things from the analysis, depending upon how we did the genome-wide association. And I have to say this is one of my perks for using SVS in changing my association analysis, is because there were a number of times where I did that association numerous different ways, whether I included the Roma sinuano or took them out, or I included the Karora or I took them out. So we analyzed them in multiple different ways to see, did our association peak change position? Did it remain the same? Did it go upstream or downstream? And what was the significance, the impact on the significance? And with that being said, the haplotype blocks in the association, when we altered those types of things, we really found that the Senapol and Roma sinuano seem to basically appear very similar, and when we put them all together, we have really good results. The Karora, on the other hand, they improve our association results, but their haplotype analysis generally doesn't agree with the Senapol and Roma sinuano. So a lot of times, our theory, at least my working theory right now, is that we have potentially two mutations affecting the same gene, and it's related to breeds. So there might be a different mutation for the Karora as opposed to the Senapol and Roma sinuano. And with this being said, we're sequencing through the region, and we're looking at genotyping additional slick breeds to try to solve those issues. So that kind of concludes basically looking at a trait association. But now we're going to jump right into African goat work and look at how I commonly tackle some population structure. So this goat work is part of a USAID Feed the Future initiative, and it's the African Goat Improvement Network. So in this particular one, you know, why is this important and why are we targeting African goats? So one of the big pictures is this is part of the USAID Food Security Initiative. So the big picture is here, we're looking at, you know, 90% of the global population is in these hunger zones, which are more of the red and the orangish areas that you see here. And our target is the African smallholder farmers. And we're looking at goats because they're one of the predominant livestock species, particularly over a lot of these hunger zones. So why else do we look at goats? You know, other than numbers, because they're some of the most common, it's also a very big livestock species that's important to smallholder farmers in particular, and this is very often women. Also, the African goats in general, they're really diverse hardy species. So as you can see in this particular goat picture, which I took in Ethiopia during sampling, you know, these goats can thrive and do well in these amazingly harsh areas where you just wonder what in the world are they eating. The other thing is that there's been very little to no selection on these animals at this time. So there's really a lot of potential growth for selection if we start breeding them for particular traits. And health status, you know, we can really have a lot of impact and improvement in selection. as well as they're economically efficient. So whereas a cattle in Africa might cost you a lot more money and have a lot more cost related to basically keeping it and the resources to keep it and manage that animal, a goat is generally, because they can survive in these really harsh climates, they're a smaller animal, they don't cost as much to obtain, and they don't cost as much to keep. So it's one that's just more economically efficient for more of the smallholders. So this is where we're at right now. This has really become a huge collaborative project. I think everyone in the project knows by now that I tend to be a very greedy person, and the more animals and samples I can get, the better. So our sampling status right now is we have goats from 16 countries. That includes over 60 sampling sites, not just countries, but villages, that type of idea. About 66 breeds or populations. So in some cases, they aren't necessarily just a breed, but we've really targeted populations if they're in a unique climate or ecotype. And in some cases, they don't have a particular breed name. They're just known as the Malawian goat. So we've really targeted as many different ones, particularly indigenous ones, that we could find. And this has included almost 3,000 goats that we've sampled at this point. So specifically in Africa, we have 11 countries that we've sampled from with collaborators, and we have 4 new FAO research projects that are being added. Each of the pie charts, so the names of the countries are the countries that are actually sampled to date. The pie charts give you an idea of the different breeds and the number of animals from each of those breeds that we've sampled. So you can see some country, it might be 1 or 2 breeds, maybe even 3 breeds, and they might be very similar numbers. Other countries, it might be predominantly 1 breed that you're seeing sampled. But because we're really targeting unique ecotypes and kind of comparing some goats across countries, we still tried to get, a wide variety of the breeds that were in that area. So I said we had sampled 16 countries, so we actually have a few countries that there's a global comparison for us. So some from the US, including the Boer goat, which originated in South Africa, but also some Spanish descent goats, as well as Italian dairy goats that are important for our analysis because the genotyping chip that I'll show you as we come forward is based on dairy goats. So that was very interesting, as well as Turkey is more closer to the domestication center. Brazil gives us a unique comparison of goats that are in a similar climate and ecotype. And New Zealand has some early imported lineages of goats from early imported South African Boer goats that we compared to. So what did we actually collect on these animals? So when we went out to start sampling, one of our first priorities was to basically make this standardized sampling protocol. So this is the first round, and you have to keep in mind that we're going out, we're collecting a lot of animals, time and logistics are of the essence here. So the way that we tackle this was we have one major factor is geographical information system, so latitude, longitude, elevation, and this can basically allow us to go backwards and look at climate data. We also did a very basic set of physical body measurements, 6 body measurements that we're looking at. We have a graduate student, Jennifer Woodward Green from George Mason University, that's doing her project on photo characterization. And in the long run, that's looking at the idea of basically being able to take a picture of your goat and run it through an analysis that'll basically give you an estimated size of the goat, the physical body measurements. And then lastly, we were looking at the biological samples. So we normally chose either blood samples or tissue samples as our biological samples for DNA extraction. And that really gives us some of the higher quality and more DNA quantity. So the outcome of some of this information, we're in the initial phases of, running and processing our samples. So thinking about those physical body measurements, one of the ideas was looking at the variation in body size. So the way these charts are made, my student Mary Beth Hannon has been working on this, and she's basically classified average body measurements, whether it's height in the top graph or length in the bottom graph. Both of these are highly correlated. And basically each individual bar is so this includes, for instance, the very first group is the animals. We only have 2 breeds in Cameroon. The blue bar is a representation of all of their kind of local crosses that they call them. And this is an average for length and height of those crosses. And then the next line would give the average height of all the West African dwarf that were sampled in Cameroon. And that's kind of how these are laid out. So it gives us an opportunity to look at, unique things such as the Boer goats and how much, they're bigger in body size versus some of the other breeds. So this is the start of our variation in body size. And kind of why are we looking at this other than just what is the variation and giving us guidelines for these breeds? Is there a significance to this, and does it range across countries or across breeds? So one way to look at this is we do have Boer goats from a number of different countries. And here you can see the different countries are each bar, and we have our 5 different measurements that we were looking at in this particular case. And some of the things that we want to point out, we're looking for where's their significant difference. So one of the things that, stood out immediately was in the lower left purple chart, looking at how much difference the Mozambique animals were in their chest width. But on the other hand, when we went back and kind of looked at the numbers, we also related that in Mozambique, we only had 2 goats. So this is definitely a skewed data set right now, but this is our preliminary one that we're going through and we're starting to average. But we can also look at unique things such as when it comes to the average weight of the Boer goat, the US definitely has the heavier goats in general. So this is one way to look at it, look at a single breed across multiple countries. Another way is to look within a single country and then look at the different breeds and how this goes. And now this data set's a little bit more balanced. Each of the breeds, there's roughly the same number of animals, and we have 2 breeds that are in the north as well as 2 in the south of Ethiopia. And now that I show you kind of the measurements, the very first column, keep in mind, that's all the breeds put together, their average. But then the next 2 bars are going to be the 2 northern goats, and the last 2 bars are the 2 southern goats. And then the unique situation here is that the 2 northern breeds not only are in the north, but they are also in more of a lowland elevation, medium to lowland elevation, versus the last 2 breeds that are in the south of the country are actually highland breed populations. So this is something, is the body size actually related to elevation and where they are, which is probably tied back into food availability in those areas. So that was kind of taking our body measurements, kind of the early data that we have, but we've also started genotyping the animals, and body measurement will come back into future analysis, genetic analysis. So genotyping right now, we've gotten goats genotyped from 11 countries and 41 breeds. We've done SNP filtering using SVS that we've basically narrowed this down from the original 53,000 SNPs on the Illumina SNP chip to about 51,500 based on our threshold settings. And this has really given us basically sample filtering using a 90% call rate for samples has narrowed it down from 952 individuals to 895. So that leaves us with the analysis that I'm going to show you. We have about 895 animals, and we're using about 51,500 SNPs for our analysis. So the project analysis, this is really laying the foundation for our project, really looking at these are the different breeds or populations we've sampled, and what is that showing us genetically? Are they actual unique breeds, or are they, how related are those breeds? So the first thing we started with was principal component analysis. So principal component analysis is really the idea that you're looking for these unique genetic signatures. So we're putting in all of these genotypes from all these different breeds, and based on the genetics alone, what is the most significant factor or pattern that's being pulled out? So this is a scatter plot, and we have PC1, or the first principal component, which is the most significant value of differentiation on the x-axis, and then PC2, which is the next most significant value on the y-axis. So for me, this is kind of where I really enjoy it. And the more you know about the breeds, the more you can kind of come out and say, this is what you think might be going on in this particular analysis. So for PC1, you'll see here that I have lots of different names of the countries and circles, kind of some of the groups circled where those particular individuals are. So each individual dot is an individual goat. They're colored right now in this diagram based on the country of origin that they were sampled from. And there's some cases, such as the Boer goat, as mentioned previously, that we sampled from multiple countries. So you'll see down at the very bottom, We have both the US and the New Zealand boar goats, as well as the South African boar goats are generally going in that direction as well. We have, to the far right in the top, we have all three of the Italian dairy breeds, and the Spanish ancestral breeds from the US are basically in that direction as well. And we have the Brazilian breeds, but they are also a composite of some of this European ancestry. And then basically the other thing I want to point out is then to the left-hand side of the chart, you really have almost all of the African countries. So one of the things is basically now we got to solve, you know, what is potentially PC1? What is that differentiating? And for me, PC1 is really separating kind of that European versus the African ancestry, the Italian and Spanish ancestry versus the African countries. PC2, which is that vertical y-axis, is differentiating really the boar goats that are on the very bottom from all of the other breeds. So instead of showing lots and lots of slides of PCA, I kind of put these together, and I put out basically the factor in that table at the top left is what I term it. You know, this is basically what I'm seeing. So from the previous diagram, PC1 is that European African, PC2 is more the boar breed itself. PC3 is really distinguishing the Turkish breeds. 4 kind of brought out more the Nigerian breeds, and 5 brought out more the South African breeds. So this is just giving us an idea of which different components and genetic signatures are significant when I use all of these individuals. So we can look at this a little bit differently. You know, the previous ones I had the European ancestry, but if I want to just look at the African countries, I might see a little bit different dynamics or see what's happening just within Africa. So these are box plots of principal component analysis, and box plots, sometimes I find it a little bit easier to look at it and decide what's actually going on in a component when I do it this way. So both of these box plots are the exact same data set. It's only African countries, but I have the individuals grouped slightly different. In the left-hand side, I've grouped all of the breeds for a country together as a country. On the right-hand side, I have it grouped in a smaller way, just by breed itself. And what I'm trying to show is, you know, it depends on how you want to see this and what you know about the populations. So in this case, if I look at it by country, it seems very clear that PC1, when I'm just analyzing this data set, is really showing the uniqueness of the South African goats. Whereas when I look at it by breed, I see that there's a couple breeds that, you know, basically go to the left versus the right. And if I know the history of those breeds, I can say all of those animals are from South Africa. All of those breeds are South African breeds. So it's basically, it's showing me the same thing. It's just a matter of to what scale are you interested in. And you can break this down. You know, principal component, a lot of the analysis we do is really, it's very sensitive to what are the actual individuals that you're putting in the analysis. So we went initially from a huge data set of European, Brazilian, as well as African animals, to now we're looking at a data set that only has 3 African countries, as well as the New Zealand boar, which were originally derived from South Africa. And here you can see, you know, it's much more clear that each different color is actually a breed, not just a country, but a breed. And you can see how, particularly in Ethiopia, as well as the Nigerian and New Zealand breeds, are very easy to distinguish one breed from another breed. As it's a little bit different, though, when you look at Kenya down in the lower right-hand corner, because in this case, you see that a lot of these breeds really overlap. And I think this shows a lot of kind of the history in some of the Kenyan goats, where there's been, you know, a number of cases where they've brought in exotic breeds, European dairy breeds to mix with the African indigenous goats. So there really is a lot of admixture going on, and that's evident in how they're all clustering one on top of the other one. So I think it's going to be a lot harder to tease out the indigenous breeds versus, you know, the admixed breeds. But it's something that through our project, when we started out, we looked at indigenous breeds, and they were kind of a focal point. But we realized there's some countries where we need to address that it's not just the indigenous breeds, but we need to look at how do we improve these admixed animals that have a very unique ancestry. Another way that we're starting to look at some of these goats is just looking at the idea of signatures of selection. In this particular case, I have a Manhattan plot, and it's based on marker-based FST. And so when you do this, I'm comparing in this data set I compared all of the African goats to all of the European ancestry goats. And I can see where in the genome am I showing basically population differentiation between those two groups. So you can see across the genome there's areas of increasing differentiation. And this is as far as we've gotten in the analysis, but it'll be an opportunity that we can go back and say, you know, are there unique genes here that maybe have something to do with parasite resistance or climate tolerance, that type of thing. So we'll be investigating that further. But this is, you know, at least a rough idea of, you know, how do the African breeds compare to some of the European ancestry. So far where we are with the African Goat Improvement Project is we've looked at basically both body size and genetic investigation does show us variation among goat breeds. And we're determining kind of the level of significance. You know, are the Boer goats in the US really significantly, do they weigh more than the others, or is it really just not that significant? As well as, you know, the genomic analysis and population structure through PCA generally reflects country and breed divergence, but we can look into basically further development of the breeds themselves, and does this reflect any migration patterns that we can see. Lastly, the other thing that we're kind of looking at right now is basically the genomic analysis demonstrates this potential for regions of the genome to be under selection, you know, when we compare the African versus the European ancestry. So all of this work with the African Goat Improvement Project is really kind of laying the foundation for these are the unique populations, these are the more related populations, and helping identify ones. Do you want to, you know, basically look at conserving this breed or moving forward on breed improvement, whether it's for body size or reproduction or milk production, or are these, you know, which ones are very unique versus very related breeds. So kind of helping the locals decide and prioritize how they want to move forward with breeding programs. So all of the projects that I showed you today really use this genome-wide investigation, and I'm a big, most of my research uses snip chips, so I'm using thousands of markers across the genome. Whether I do trait association or I'm directly looking at population structure, I always do some of that background anyways to get a more appropriate data set for my research project, looking at the relatedness of individuals, including inbreeding, unique genetic signatures, or selection for conservation or improvement. When I move on to do association studies, I'm really trying to identify these regions of the genome that are in association with whatever trait of interest I have, and looking at, you know, what's the genetic regulation of that trait, as well as I'm a very applied person, so I also like to look at identifying diagnostic markers for most traits. For all of this work, within my lab, Mary Beth Hannon's been working on the GOAT project with me. The Thermotolerance project is a fairly large collaborative one across many groups bringing in different information, as well as the African Goat Improvement Network has a hugely collaborative project. And here I could fill up multiple slides on all the individuals in this particular project, so I elected for taking the easier route and putting the larger groups that are on there. But all of this is very collaborative work, and I think most of what I do nowadays, I really rely on that and appreciate that. Lastly, I'd like to put it out and appreciate any questions, and I'm happy to answer whatever I can from you.
Thank you so much, Heather. As Heather mentioned, we're going to go ahead and answer some questions. If you haven't had a chance, feel free to type them into the questions pane. I have a couple of quick announcements on our side. I will have the webcast recording up on our website tomorrow at the latest. Our next scheduled webcast is on August the 6th, and it will cover some tips and tricks for using the SVS software. We will be sending out invitations in the next couple of days. With that, Heather, why don't we go ahead and answer some questions?
Oh, I just looked at, and I know exactly where this is coming from, ROH blocks. So one of the questions is the size of the ROH blocks that Isu Kim ran during our SLIC analysis. He used, in that particular study, what he did was he did basically looking at different block sizes. And in his case, the analysis tool he had, I believe he ran it in R, and he was looking at basically blocks of different 100 to 500 base pairs. And I think we were really trying to use that as going backwards and using it part of our haplotype analysis as well, looking for the runs in seeing the difference between the centipole versus what were the runs that we were seeing in the ancestral breeds. And then, so we would compare like the centipole to the centipole Holstein crosses and the Holstein themselves. And then we'd go back in and we would look at haplotype blocks that were in the Carora as opposed to the brown Swiss. So I have to say that initially it was just the way that we approached it, but you could probably go back and look for smaller blocks and see if it altered where the pattern was. Are there any other questions? So the one other question that just came in was, what was the major software that was used in the analysis? When we did the SLIC project, that one we used a multiple of projects. So we utilized SVS for the original association. I used Emacs. At that time, the mixed linear models was not part of SVS, so I would export my data and put it in Emacs directly for the other GWAS. I have to say now I'm doing projects, and I love the fact that it's all in one thing, so I do it all in SVS. The other thing, I used HaploView for the haplotype analysis, the haplotype blocks, looking for actual allelic patterns. I used HaploView. The PCA was done all in SVS. I have to say the majority of the stuff that I do right now, I use SVS. What's the next one? So there's another question that just came in about the genetic signatures for the Turkish breeds. And I have to say, in this particular analysis, I'm using the Turkish breeds more as a reference population to see where the African breeds relate to the Turkish breeds, as well as, you know, kind of making sure that things kind of fall in line between the European ancestry. There's an independent project that's going on with our collaborator that's looking specifically at the Turkish breeds. So she'll be analyzing that data. I'm sorry, folks. I'm reading questions as we go. They're appearing on my screen. Okay, so there was one. Let me try to go backwards to the PCA. Okay, so one question was not quite understanding, I think, these two slides. So a particular question was, I think the idea is just the difference in these two, the slides that are here. So they are the same data set, so the exact same animals are being analyzed. But what I did, so the PCAs and values for individual animals are the same. But when I did the box plots, I put all of, so for the one on the left by country, I would basically put all of the individual animals that are from Uganda. Didn't matter on the breed, but every animal from Uganda was included in basically the clustering for Uganda. So by country, every goat that I had for that country is averaged together for the plot on the countryside. When you look at it by breed, instead of saying what country they came from, here I clustered individuals based on the breed that they belong to. So it's just different ways to look at it. So to me, in this particular case, I can say PC1 seems to really be differentiating South Africa versus all the other countries. So it's the goats from South Africa versus the other countries. Whereas by breed, if I look at that one, I can say, so here I have to say that I have these broken down into 3-letter abbreviations most of the time. So I can see that there's a unique breed right at the top, the TWA and the SVH. So that's the Tankwa and the Savannah breeds. And so I can say, well, those are two breeds that are, you know, definitely being segregated, along with the other ones that are on the left are being segregated by PC1. If I didn't know the history that all of those breeds that are on the left happen to come from South Africa, then that would be something that I'm just saying, you know, here are unique breeds that are more related or have a common principal component 1. Whereas if I differentiate it by country, it stands out right away that these are all from South Africa. So it's just different ways to look at it and kind of help solve the puzzle. So one question looks at sample size, particularly in the goat project. So as we set up for the African Goat Improvement Project, because we were trying to get such a large data set and sample so many countries and as many populations as we could, generally our threshold is we're really trying to pick at least 25 to 30 individuals to represent every breed. There's a few unique breeds that, you know, if they're in a very unique ecotype, and we could still only sample a few animals, you know, where you run into challenges is you might go to this unique ecotype and maybe there's a population of 300 animals, but if they're all pretty much very related, it's going to skew your genetic analysis. So even though they have 300 animals, you might only sample, you know, 20 individuals from there or 30 individuals or so on. You really want to try to get, you know, different farmers, different populations of those same animals. So at least the way that we've set it up for this particular project, we're generally shooting for 25 to 30 individuals that we genotype to represent each breed. But I will say that when you're looking at admixture, NPCA, all of those, you know, they are sensitive to the number of samples you have. I try to balance out my samples the best that I can. So when I showed the Senepal ancestry, you know, in there, I'm definitely biasing the one Senepal ancestry because I had 38 Senepal animals, and I only had 10 Ndama, 10 red pole, 10 zebu. So this is skewing it to the fact that I'm swamping my data set with more Senepal than anything else, so it's more likely to bring out that Senepal signature. You know, if I just wanted to do a breed analysis, I would just do 10 of each breed or 20 of each breed. In that case, I chose to keep all the Senepal because I was really interested what was the variation from one Senepal to another Senepal. So that case, I wanted to look at every individual Senepal in my analysis. So that's why I did keep it that way. But if I were doing more of a population study, then I would balance out each population the best I could with about the same numbers. There's a question on, do we have the same standard for the measurements from the goats from different countries? I'm not sure if I'm quite understanding it right, but I guess the way I'd answer that is that we really did make the standardized phenotyping protocol before we started the project, and we tested that out basically on local goat farms. So we really did basically, when we put this out to collaborators, they all got the same protocol, and particularly in the very beginning, what we did was myself, I went over as well as the graduate student, and we worked with our initial collaborators, Hans Solkner and his graduate students in Ethiopia, as well as ILRI researchers in Kenya. So basically, we were there together to teach them, you know, these are the measurements, where we're measuring, how we're measuring, these are how we're taking the photos, even the sample collection, but that was pretty straightforward. So we basically all worked together. We developed it as a standardized protocol. We taught the other groups how to measure it. We had Azareka as an affiliation, a group in Africa, do the sampling in a number of our countries, and even their lead researcher came up and studied with us when we sampled in Kenya. So he saw, again, we were able to do the training there of this is how we measure the goats. During our, we've had annual workshops for our African Goat Project, and those have often been, you know, updates as well as training. So during our second workshop, we had a number of countries that were kind of new to the project come in, and we actually did a site demonstration where we showed all of our collaborators how we were sampling the goats. So we tried to make it as standardized as possible. I will say that, you know, even with that said, we've still had, you know, a country or two where we've had data sets come back and we're like, you know, something went wrong and we can't include, you know, this pin bone measurement. So we've made it as standard as possible with training, but there's still human error. How did I make the box plots in PCA? R. I actually used R as the program. So I ran PCA and SVS. I exported my PCA values into basically a text file, and then I put them in R, and I use Jaguar because I kind of like that interface versus just the command lines, and I did the box plots there. There's another question, the marker-based FST, the one that I showed was African versus European, and yes, we will definitely be looking at that, I think there's a number of different ways we can do that. We can do it based on ecotype, we can do it based on elevations, so yeah, I think there's a lot of different ways, and the more that we can get information on each breed, the more we can figure out different ways to kind of analyze that and probably find unique signatures. There's a question on using the pictures to come up with the body measurements. So our graduate student is actually doing really well on this. She is in the midst of right now analyzing the photos. So right now she's at the part where for our initial setup, you know, she really had to do a very intensive thing where we had to have blue tarps and blue background, and the person had to be holding the goat had to have like a blue coat. We assumed that there's no blue goats, so that's why all the background's blue. But we really went on the idea that, you know, differentiating the goat as much as possible from any background. And she is currently writing algorithms so that, you know, how to basically, in a photo through computer programs, how to isolate the goat itself and look at, you know, finding out those body measurements. Right now she's evaluating, you know, ways to basically from the pictures generate the most reliable estimate of body weight and height and length. So she is working on that. It's improving, but it's still, that's part of her project, so it's in the process. One of the questions was actually, is there any correlation between slick hair with sweating rate on the cattle? And I don't know for sure if I don't know that there's specific correlations that have been done between the sweating rate, but I know that some of the research has actually looked at, there's a couple other papers out by some of our collaborators and other folks that have looked at more the hair follicle, the size of the follicle, as well as the number of follicles. So that information, there is research out there that it is correlated with some of those and giving averages of slick hair coats versus more normal hair coats that we're used to. There's one question about, can the snip panel in the goats, well, I guess I was assuming goats, but I'm not sure if they're referring to cattle or goats, but can the snip panel be used for analysis of breeds all over the world? And my opinion is absolutely yes. So I've done cattle, I've done goats, I've looked at dogs, and all of those I've used Illumina whole genome snip chip data. I will say that the more closely related the breeds that you're investigating, the more SNPs that I would recommend, that the SNPs can really have a different, an influence on kind of teasing out, you know, unique signatures in closely related animals. However, you can break down a SNP panel to very few SNPs and still find the same big picture. Particularly in cattle, you can break it down. You know, if you run PCA or even admixture, whether you're using 700,000 SNPs, 50,000 SNPs, or 3,000 SNPs, the first couple things you'll find out is you'll immediately break down Taurus versus Indicus, then you'll bring out the African cattle. So some of the major signatures that you get are generally going to be the same, but it does, you start to see more of an influence on the number of SNPs when you have more closely related breeds. So yes, I use these SNP panels all the time to compare breeds. There's a question, is the population structure, so I think we're looking at the goat project. I think they're asking, did I remove any animals based on admixture types? So the graphs that I showed you, I did not remove any animals unless they didn't go through qualify because of quality control. So such as they didn't have a good genotyping call rate. But in this analysis, I didn't remove any of them. And even when I did the Senepol cattle part, You know, if there was a kind of a bizarre situation where one centipole, you know, was much more, showed a different admixture than the other group, I left it. I mean, that was still a centipole. So, and for the project that I was doing there, you know, that didn't have a huge impact. I have to say that personally I'm very leery about taking animals out of my analysis just because they don't fit the data set that I want them to. So I very rarely take them out unless I honestly can go back and say that I think there was a genotyping error or a sample identification error. So most of the time I don't take them out unless I have a very positive reason for saying that this animal really shouldn't be in the data set the way that it is. There's a question on how close are we to the SLIC mutation. It depends what day you ask my colleague, Tad Sonstergaard's the one that's kind of leading that part. And yeah, there's good days and bad days, but we're working on it. So hopefully that'll be coming out in our next publication. But yeah, we are definitely getting closer to it. And hopefully it's right now it's more on the verification with new SLIC breeds and SLIC animals that we're genotyping. Oh, an update here. For population analysis, you know, there is the comment that kind of, as I was mentioning, so one question had been, can you use the snip chips or this snip data for population analysis? And I said, yes, that I do that commonly. If you are doing that in cattle, well, in any breed, you do have to look at, you know, what are potential biases. For the goat work, we looked at, you know, the chip was based on more of a dairy breed, and that's why we have some dairy background in there that we're comparing to. For the cattle nowadays, really the high density one has both Indicus and Taurus chips, and, you know, really looked at going across populations. So the high density one's probably the one that was made specifically to go across breeds. But yeah, so you do have to keep those in mind when you're doing any of the population analysis. Another question was, what type of sample did we collect for DNA extraction from the goats? So we generally we always collected 2 samples per animal, just as a backup. So we normally collected either 2 tissue samples using ear tissue punches, or we collected a tissue and a blood sample. We've had some troubles with the blood samples just logistically in Africa because of the heat and transport, that type of thing. Number of people have used hair samples. We opted that we can get the tissue samples and blood, and tissue has really proven the best one for us for the African project. And we get more DNA and higher quality DNA because some of this we want to keep not only for SNP chips, but for the potential for whole genome sequencing. So that's why we really wanted to have the higher quantity and quality. So you can use hair samples as long as people know that, you know, you got to get the hair bulbs and get a number of hair bulbs. But you just don't have as much DNA to work with then. I think I've hit most of the questions that have come in. I seem to get new ones like every moment.
Thanks, Heather. We can go ahead and wrap, and as they come in, I will make sure that I get them to you for answering, and then we can get the answers to the questions that you've asked. Excellent webcast. We really appreciate your time, and we appreciate you sharing your research with us. We're going to close now. I wish everyone a fantastic day, and we will see you next time.
Thank you.